Hello there. In this video, I'm going to talk about the cell cycle checkpoints. So in other videos, I've been discussing about the interphase and the mitosis phase, which are the cell cycle processes which leads to cell division. So during the same cell cycle, there are what we call the checkpoints, which checks the cell for various processes. So by definition, the checkpoints are the points which holds the progress of the cell to the next stage if the cell is compromised. In other words, we can say these are the points which stops the progress of the cell to the next stage if the cell is damaged or compromised. Okay, so during the cell cycle, we said we have this phases like the G1 S phase and G2 phase, and we also have what we call the mitosis or the mitotic phase. So during this phase, we have checkpoints where the cell is checked for various reasons which we are going to discuss in this video. So the first checkpoint is the G1 checkpoint. Then the second checkpoint is the G2 checkpoint, which is there, and the last checkpoint is the M checkpoint, which is the last checkpoint in the cell cycle. Okay, so let's start by looking at the first checkpoint, which is the G1 checkpoint. So under the G1 checkpoint, the cell is checked or is assessed whether the requirements needed to proceed to the S phase are met. Under the G1 checkpoint, which is this checkpoint here, the cell is checked whether the requirement to proceed to the next phase, which is the S phase, are met. So that's what is checked under the G1 phase. Another thing is that the DNA genes are also checked if they are in good condition. The DNA genes are checked as well, and the cell is large enough. So the G1 checkpoint also checks if the cell is also large enough to proceed to the S phase. Then from there, we have the G2 checkpoint. Now under the G2 checkpoint, which is this checkpoint which is here near the mitotic phase, okay, so that checkpoint there checks the cell whether the cell has replicated, the DNA has replicated. Remember, under this S phase here, the DNA is replicated. So as it proceeds to the G2 phase, it will be checked to see if the DNA has replicated. Okay, and then from there, the G2 phase also checks the environment, if it is suitable for the next phase. It checks whether the environment is suitable to proceed to the next phase. So that's what the G2 checkpoint does. Then from there, we have the M checkpoint, which is the last checkpoint in the cell cycle, which is this checkpoint here. So that checkpoint there, it ensures that the sister chromatids are correctly attached to the spindle microtubules. As we discussed in our previous video under the mitotic phases, there is what we call the attachment of the spindle fibers to the chromosomes at the region called the kinetochores. So this checkpoint now, it checks to see if the spindle fibers have correctly attached to the spindle microtubules. So that's what the M checkpoint does. Okay, so from there we have what we call the protein p53. So this protein, what it does, it is the one which is responsible for the cell to progress to the next phase. It is the one which regulates the checkpoint. Okay, so this is the internal signaling protein, and it is called the cyclin. It is called cyclin. So these cyclins, they are responsible for the progression of the cell through various checkpoints. So it helps the cell, or it signals the cell to proceed to the next checkpoint. Why is this p53 important, and what is its function? So, but before we look at that, let's also know what this term means. This term hypoxia. So this is the condition of reduced oxygen supply. Is simply the condition of reduced oxygen supply. Okay, so let's look at what this p53 does. So this p53, what it does is that as the cell proceeds from different checkpoint, the G1 checkpoint, G2 checkpoint, and to proceeds to the mitotic checkpoint, it what it does is to stop or to signal the cell if there is any problem, if the cell is not in good condition, like if the cell has an abnormality, if there is an abnormality in the cell, then p53, what it will do, it will arrest the cell, it will stop the cell, and then that damage in the DNA will be repaired. After that the DNA has been repaired, the cell will proceed to another stage of the cell cycle, or the cell cycle will restart. So that's what it does. It checks if the DNA is correct, or it has some faults in some way. Then it will cease, or it will stop that DNA from to proceed to the next stage of the cell cycle, and it will signal the repair mechanism. So the cell will be repaired and the cell cycle proceeds. But what happens if the DNA mechanism has failed? So if the DNA damage has failed, then it will signal another process which is called apoptosis, the programmed cell death. So it will signal the apoptosis, which is simply the programmed cell death, and that cell will die. So that's what this p53 does. Now let's imagine that this p53 is damaged as well, or let's say it is not functioning properly. What will happen? So what will happen is that if this p53 is damaged, the cell cycle will continue with the damaged DNA, with compromised DNA, and what will be the result of that will be cancer, the problem which we call cancer. Now the cell will continue to replicate a damaged DNA continuously, which will lead to what we call cancer. Okay, so that's all in this video. Thank you very much for watching. Don't forget to like and subscribe to my channel so that you don't miss any of my videos, and don't forget to drop your comment under the comment section below. And if at all you don't like my video, please just hit that unlike icon down there.